Okay, freshmen, good morning. It is our fourth day of the genetics unit. It's Monday, March 30th. We're now in our third week of virtual learning. I know for me, I'm starting to feel a little bit more like I'm in a routine, hopefully for you guys as well. Remember what I talked about, I think during the Zoom last week, that getting outside once a day, doing something active once a day can be really beneficial in terms of your mental health. I would argue that your mental health, staying calm, staying not depressed, not anxious is more important than, Punnett squares and whatever you're learning in your other classes. So just make sure that you guys are taking that seriously as well. As I mentioned in the Zoom last week, for those that are able to make it, we will plan on doing another Zoom session this week, Wednesday at 2:30. So you should finish 2 days of lecture. That would be Monday and Wednesday, and then Monday afternoon at 2:30. Anyone who has questions can jump on. We can do more practice problems. If you just want to check in and say hi, that is absolutely okay. So from last week, the last thing we learned was multiple alleles for blood types. And so we did the first 3 questions of the packet. Today, we're going to learn one more complex pattern of heredity, which is sex-linked or X-linked traits. And then your homework is to do the last 2 questions on the genetics packet. So those of you that were in the Zoom last week got the answers to these first 2 as we discussed it. We have 2 people with blood type B. They're both heterozygous, but with multiple alleles. Since B dominates over I, these individuals have blood type B. We do the cross, and one thing we notice here is that we have 3 individuals, not 3 individuals, but 3 outcomes out of 4, so a 75% chance of the offspring being blood type B. But realize that even though all 3 of these that I just drew are blood type B, only one of them is homozygous dominant. The other 2 are heterozygous. So you need to realize that these 3 all have the same phenotype, but they have different genotypes. Then this last box is going to be genotype II, phenotype type O blood. So our genotype ratio, I think the easiest way to do it is just label your answers. We have 1BB. Try to write in order from most dominant to least dominant. It just kind of is a good pattern to keep. We got 2BI and 1II. Now for phenotype ratio, because these 2 have the exact same phenotype, it's a 3-to-1 outcome. So again, please don't get confused, and the phenotype is not II, the phenotype is O. Don't get confused here though that this doesn't mean that this couple had 4 babies and 3 of them are type B and 1 is type O. This is just showing the probability. If this couple had a baby, there's a 75% chance the child would be type B, and there's a 25% chance the child would be type O. So again, just remember that Punnett squares are all about predicted outcomes, not actual outcomes. Okay, next one, AI and BI. Again, if you did the Zoom, you can fast forward this because you already have the answer. This one's a pretty interesting one because this is the only combination where if these 2 parents got together, had a baby, they would have equal chances of all 4 types of blood. 25% chance AB, 25% chance of B, 25% chance of A, and 25% chance of O. So the phenotype ratio, I don't need to even label because 1, 1, 1, 1, there's only 4 outcomes, so obviously each outcome has a 25% chance. The genotype ratio I should label because even though it's still 1, 1, 1, 1, remember that there's 6 possible genotypes, and so 2 genotypes, A, and B, are not present. So for genotype ratio, if I just wrote 1, 1, 1, 1, it would not be clear what we had. So 1AB, 1AI, 1BB, 1II. Sorry for my horrible handwriting. It's tough using the mouse on my laptop. If you're grading, half a point for genotype ratio, half point for phenotype ratio. I don't actually grade the Punnett square because you had to do the Punnett square correct to get the right answer. All right, and then the last question for blood type. We have AB, we have II, which is the genotype for blood type O. It looks like there's only going to be two outcomes. The individual is going to be BI or AI. So for genotype ratio, let me check my answers. Yep, that looks right. 2 AI to BI. And phenotype ratio 2A to B. 50-50 chance there. Okay, so just 3 points from the homework from what we've done so far. Okay, getting into class today, we're going to learn about sex-linked traits, also known as X-linked traits, so go ahead and get out your notes. This is the fourth type of complex heredity. X-linked, I've always called it sex-linked, but the IB refers to it as X-linked because it does happen on the X chromosome, so just know both of these terms. But basically, it's a trait that's carried only on the X chromosome, and they're recessive. So everything we've learned about so far is considered autosomal, so that's a term you should know. If I ask you for a autosomal trait, I'm talking about anything we've learned about thus far, because those are all happening on the other 22 chromosomes. But anything happening on that 23rd X chromosome is called an X-linked trait. An example of this would be hemophilia. Now here's where it gets a little interesting. A man who is hemophiliac would not have homozygous recessive H. He would just have one recessive H, and that's because men only have one X chromosome. So you only have one shot to have the disease or not have the disease, where a woman has two X chromosomes. So if a woman had this genotype, X dominant H, X recessive H, she would not have it because the dominant trait would be to not have hemophilia. Because of this, men are much more likely to have sex-linked traits because they only have one X chromosome, so they only get one chance to have that healthy allele. The example that we're going to talk about in a moment is color blindness, and you may already know this, but men are much more likely to be colorblind than women. So one thing you do need to remember is the chromosome difference between men and women. We can do a Punnett square just for chromosomes, and it makes sense just looking at the offspring of a man and a woman when they have a baby. So the woman's here. A woman 100% of the time passes on her X chromosome. What that means is that women do not determine the sex of their child. So those of you that are male, those of you that are female, it's because of your dad. It's because your dad has an XY chromosome. 50% of the time, the man passes on his X chromosome. And if the resulting offspring is XX, it's female. 50% of the time, men pass on their Y chromosome, and that result is a male. I have two daughters, so what that means is that both times I reproduced and my cells went through meiosis, I just happened to pass on the X chromosome. It was a 50-50 chance each time. There's a misconception out there. A lot of people will say, oh, use me as an example. I have two daughters. If I ask the average person, if I have a third kid, am I more likely to have a son or a daughter? Most people will say, oh, you're more likely to have a son. But that's incorrect. Again, this is likelihood of offspring. Every time you have a child, it's 50% chance of female, XX, 50% chance of male, XY. So if I had another baby, it would be a 50-50 chance of being male or female. But now we're going to attach some traits to these chromosomes. And so the reason why this is a bit complicated is because we're looking at sex outcome, male or female, and the trait outcome. So it's sort of doing two things at once. So the trait we're going to study for this one is color blindness. Capital B, or dominant B, represents normal color vision. Recessive B represents color blindness. So realize that the letter doesn't actually matter. It's more about the capital or the lowercase. The capital showing dominant trait, you see normal colors. The lowercase being recessive trait, meaning you're colorblind. People who are colorblind don't see in black and white. It mostly means that red and green end up looking the same to them, but they do see most colors. All right, so here's the genotype and phenotype possibilities. For females, there's 3 outcomes. You could be homozygous dominant, which is normal vision. You could be heterozygous, which is normal vision, or you could be homozygous recessive, which is colorblind. For males, there's only 2 outcomes because there's only one X chromosome, so you either are dominant or recessive, you're either normal or colorblind. And this is something you guys have probably heard of before, but maybe never looked at it like mathematically within a Punnett square. This female right here, dominant B, recessive B, meaning heterozygous, she's considered a carrier. And what that means is that she carries the trait for the disease, even though she does not have the disease. So this woman could have a offspring who's colorblind, even though they do not carry the gene. Whereas this woman is not going to have a colorblind offspring because she's not a carrier either. Okay, so you have 5 genotype outcomes and 4 phenotype outcomes. Same with multiple alleles, you should label the ratios. You should be able to write them down, but from the previous slide, but the genotype outcomes for female would be heterozygous, homozygous dominant, and homozygous recessive. That's 3 outcomes right there. And then for male, it's either dominant or recessive, so 2 outcomes there, 5 total. Phenotype, there's 4 outcomes. A normal male, a colorblind male, a normal female, or a colorblind female. So you got 4 possible phenotype outcomes. Realize that the one genotype that does not have its own phenotype is a heterozygous female because she's a carrier, she has a different genotype, but she sees normal colors, so she has the same phenotype as a homozygous dominant female. These last 2 slides get a little complicated, so I might rewind, rewatch it, double check your notes, because if you get lost on this part, it's going to be hard to correctly do the Punnett squares. Okay, so time for our first practice. Again, like always, doesn't matter if you put male or female on top or bottom. I like to use superscript, but you can use subscript as well and put them like low down in the letter. It doesn't matter. Just make sure that you're also including the sex chromosome in your answer. Okay, so we're crossing a normal vision male with a colorblind female. Obviously, there's a 50% chance that they have a male and a 50% chance that they have a female. So sometimes I just like to go through and fill out this first part before I even look at the trait, just to remind myself that there's really 2 outcomes, male or female. As far as the traits, if this couple has a daughter, remember XX, she will definitely 100% have normal color vision, but she will 100% be a carrier. If this couple has a son, he will be colorblind. 100% chance. And again, the reason why, only with one X chromosome, the male gets his X chromosome from mom, because any male out there, you got your Y chromosome from dad, that's why you're male, and you got your X chromosome from mom. And so any male out there that's colorblind, it's because their mom was either a carrier or was colorblind. And so as far as the ratios go, we got 2 heterozygous females, colon, 2 recessive males. For the genotype ratio, I'm going to use F for female, M for male. I'm going to use N for normal and CB for colorblind. So phenotype ratio to normal females to colorblind males. So in this exact trait, it's pretty interesting. If this couple has a boy, we know right away he's going to be colorblind. If this couple had a girl, we would know she would be a carrier for colorblindness. Okay, one more question. Go ahead and pause the video, read the question, and try your best to answer it. Okay, so we have a female with normal vision. So here's her two possibilities found in her gametes. We have a male who is colorblind, so he has the recessive allele. What are the chances that they have a colorblind boy? So the top half of the Punnett square is where the female would end up, so I'm not even going to fill that out. I know that that's 50% of the possibility. Because the female is homozygous dominant, she is definitely giving a dominant allele to her son, so there is no chance that she would have, or that this couple would have a colorblind boy. So the answer there is A, no chance. Okay, another question. Go ahead and pause the video, read it, try to answer it. I would do it like write out the Punnett square in your notes instead of trying to answer in your head, but pause it, answer it, and then I'll give you the answer when you press play. All right, we have a male who is normal color vision. We have a female who is a carrier. What are the chances they have a colorblind child? So here we're not looking at just male or female, we're looking at colorblind child in general. So this girl is homozygous dominant. This girl would be heterozygous, meaning she's a carrier, but not colorblind. This boy would be normal, but this boy would be colorblind. So our answer there is 25% chance. Now realize you gotta be really careful with the wording on the question here. Here's where I'll try to trick you on an exam or a quiz. If I said, what are the chances they'll have a colorblind boy? The answer would be 50%, because if you look at the two outcomes, I'm sorry, if the question was, if this couple had a boy, what are the chances that he's colorblind? It would be 50%. If the question was, if this couple had a girl, what are the chances she's colorblind? It would be 0%. But since the question is, this couple has a child, we don't know if it's male or female, what are the chances it's colorblind? 25%. So just be really careful, when you read those questions, underline, annotate, make sure you're following along clearly. Okay, let's go through a couple more types for inheritance. This does not have Punnett squares linked with them, but we went through these four. A fifth one, polygenic, is when many genes determine a single trait. I've told you this a lot throughout the semester, that we often oversimplify biology so it makes sense, so it's understandable. But in reality, like eye color, it cannot be solved with a basic Punnett square. There's a lot of different genes that determine eye color. There's multiple alleles for eye color. And so in reality, a lot of our genetics cannot be solved with Punnett squares. A sixth type, environmental factors. The fact that your surroundings can influence your genotype, or your phenotype, and sometimes your genotype. An example, an Arctic fox turns from white to brown in the summertime. Flowers bloom earlier because of global warming. And then there's a subsect of environmental factors that's really interesting. Oh, here you see identical twins, different lifestyles, so clearly their phenotypes are different because of their different environments. Looks like this woman maybe worked outside her whole life. This woman maybe works in an office or inside. But that subsect of environmental inheritance is epigenetics. Epigenetics is a really interesting and more recently understood aspect of genetics. It's heritable changes in gene activity that are not caused by DNA. Basically, what's happening is things that you do, things that you eat, environments that you're in can actually change what part of your DNA gets read by mRNA and what parts get ignored. And so it's actually going to change the traits that you have. It's an idea that environmental conditions can lead to different genes passed on to the offspring. There's a pretty interesting article, Astronaut's Gene Expression No Longer the Same as His Identical Twin. It has nothing to do with him being in space, but he was under a lot of stress, and he had a very unique diet when he was orbiting, and so it changed the parts of his DNA that get read. You remember learning about histones and our DNA being wrapped around here. I would look into this. Those of you especially that are looking for something to do, those of you that are a bit bored on this virtual learning, check out epigenetics. Start with the Wikipedia page, look up some epigenetics articles, epigenetics TED Talks. There's some really interesting stuff out there that you guys could further investigate. Okay, so that's it for today's notes. The big thing was Punnett squares and sex-linked traits. So your only homework is to finish the genetics packet, page 4. Okay, I hope that was helpful. Reminder that we will plan on having a Zoom session Wednesday at 2:30. I will have a lecture posted Wednesday morning, so you guys can do day 5 before the Zoom, and then we can chat then. Okay, have a good start to your week, and I'll see you guys on Wednesday. Good morning, freshmen. It is Wednesday, April 1st. Happy first day of April. We are going to grade the rest of packet page 4. We are going to learn dihybrid crosses based on the name. You can probably guess that it's when we cross 2 traits at once. And then you do have some more homework tonight. It is page 5 and 6 of the packet. Page 5 are 2 examples of dihybrid crosses. Page 6 is meant to be fairly challenging. Page 6 is page 6 is a challenging sort of word problem based on dihybrid crosses. Okay, so the notes on dihybrid crosses. Basically, a dihybrid cross is showing the crossing of 2 traits at once. It's a nice way to mathematically show the independent assortment of alleles. You know, if I have free earlobes and a bent pinky, there's no rule that says I'm going to pass on both dominant traits or both recessive traits if I'm, say, heterozygous. But remember that the law of independent assortment does not apply for traits on the same chromosome because chromosomes do not get split. They either get passed on or not passed on. So each parent has 4 allele combinations because you have 2 outcomes of one trait, 2 outcomes of another trait, so you have 4 total outcomes. You use the mathematical acronym of FOIL, first, outer, inner, last, to write out each parent's 4 combinations. You're going to have 9 different genotype outcomes. You're going to have 4 different phenotype outcomes. If you want to try to write these down quickly, the genotype outcomes, I'll try to do them off the top of my head. Dominant, dominant, dominant heterozygous, heterozygous dominant, heterozygous heterozygous, heterozygous recessive, recessive heterozygous, recessive recessive, dominant recessive, recessive dominant. So there's 9 possibilities. I can never find the camera. Phenotype outcomes are pretty straightforward because you only have dominant and recessive. Dominant dominant, dominant recessive, recessive dominant, recessive recessive. You do not need to label these. Just try to write them in order from most to least dominant. And if you have the ratios correct, or if your numbers are correct, I'll assume that you did the Punnett square right, because the odds of you doing something wrong but still getting the right ratio are pretty slim. And again, the labeling of 9 genotype ratios is pretty intense, so we don't need to do that. Okay, so we're going to go ahead and practice our first dihybrid Punnett square. We'll use Mendel's peas as an example. So I want you to cross short heterozygous purple pea plants with heterozygous tall white pea plants. You could pause the video right now and try to write in the genotypes for both parents. And it would look like this. Again, if a pea is short, you know it's homozygous recessive. Heterozygous purple, we know what that looks like. Heterozygous tall, and then a white pea plant must be heterozygous recessive. So the first thing we do is FOIL. And remember, mathematically, it would look like this. So we have our first set of alleles. We have our second set of alleles, and this is for that first parent. I'll try to color code this. The first thing that we do is the first alleles, and that would be recessive T, dominant P. The next thing that we do is the outside alleles, and that would be recessive T, recessive P. And then we have the inner alleles, which would be recessive T, dominant P. Lastly, we have the last alleles, which would be recessive recessive. Okay, next we'd want to FOIL the other parent set of alleles. We have heterozygous for height, homozygous recessive for color. So the first alleles, the outer alleles, the inner alleles, the last alleles. Okay, next comes the tedious part of all this, where we need to fill out our squares. So what I would do, see if I can move this. Nope. What I would do is fill out one set of alleles at a time. I just think it's quicker to do that. And so I'm going to fill out all of the height alleles first. Always write, for any heterozygous, always write the dominant allele first. If this part makes total sense to you, feel free to fast forward until I get done and start adding up the genotype and phenotype ratios. But otherwise, feel free to watch as I fill this out. Why am I writing P? It's a mistake. Oops. The last box, which you can't see, is going to be the same as the second to last box, so I'll just remember to count that one twice. Okay, next, again, I'll try to color code this. I will go ahead and fill out the second allele for color. Looks like this whole column is going to be heterozygous. This whole column is going to be homozygous recessive. I'm going to redraw these because I kind of made them medium size and I might get confused if they are uppercase or lowercase. This whole column is going to be heterozygous. And this last column is going to be homozygous recessive. Remember that last one is going to look the same as this one. I can tell because these bottom two rows are the same, and it's going to be from this column. Okay, now comes the counting part, and this can get tricky. I use a system where I put X's in the boxes to annotate whether I've counted one or not. So remember, there's 9 genotype outcomes. The first one being all homozygous dominant. I don't see any that are homozygous dominant. You don't need to include zeros for this. Again, it gets kind of messy. Next would be dominant T, heterozygous P. I don't see any of those. Next would be heterozygous T, dominant P. I don't see any of those. Next, I see a lot of heterozygous T, heterozygous P. 1, 2, 3, 4. Okay, what about heterozygous T, recessive P? 1, 2, 3, 4. What about homozygous recessive T, dominant P? I don't see any. What about, oh, this should be a P right here. What about homozygous recessive T, heterozygous P? I see 1, 2, 3, 4. Last but not least, I see 1, 2, 3, 4 homozygous recessive, homozygous recessive. Notice my genotype ratio, 4, 4, 4, 4, adds up to 16. We have 4 boxes on the outside, so 4 times 4, there's 16 outcomes. So one way to check this is that your phenotype genotype outcome adds up to 16. If it doesn't add up to 16, you made a mistake somewhere. All right, next is phenotype. And here's where I'm going to continue counting by crossing out the X. So this half X shows me that I counted each one once, and I did that right. When I cross it out like this, it'll show that I counted it again for phenotype. So the first phenotype I'm looking for is dominant dominant. That would be tall and purple. Even though this box is heterozygous, the phenotype would still be tall and purple. So there's 1, 2, 3, 4. Okay, what about tall and white? 1, 2, 3, 4. What about short and purple? 1, 2, 3, 4. What about short and white, having both recessive phenotypes? 1, 2, 3, 4. So this is actually kind of a rare one where the genotype and phenotype are equal. They're both 4, 4, 4, 4. All right, another one. Short, heterozygous, purple, crossed with heterozygous, tall, and white. Pause the video, try to come up with both the parental genotypes and try to make the cross the parent outcome along the top and along the side. Okay, the genotype should look like that. When we FOIL, first, outer, inner, last. First, outer, inner, last. Okay, again, same deal. I would pause the video here, try your best to fill in all 16 squares and then fill out the genotype and phenotype ratios. I'm going to draw the 16th box over here. Just because I can't fit it. Okay, incredibly messy, but those are my 16 outcomes. I'm now looking for genotype and phenotype ratio. So I'm going to cross these out one at a time. Genotype, I don't see any that are all dominant, but I do see some that are homozygous dominant for height and heterozygous for color. So that would be 1, 2, 3, 4. I see some that are homozygous dominant for height and homozygous recessive for color. 1, 2, 3, 4. I see some that are heterozygous for height, heterozygous for color. 1, 2, 3, 4. And I see some that are heterozygous for height, homozygous recessive for color. So genotype, again, 4, 4, 4, 4. I think phenotype is going to be different here because I don't see any recessive for height. So let's just look at dominant, meaning tall and purple. 1, 2, 3, 4, 5, 6, 7, 8. And now let's look at tall and white. 1, 2, 3, 4, 5, 6, 7, 8. None that are short or purple, none that are short and white. So we see phenotype ratio 8 to 8, genotype ratio 4, 4, 4, 4. Okay, that's it for today. Your homework is the packet, page 5 and 6. As a reminder, page 5, pretty straightforward, just the practice that we just did. Page 6 is using deductive reasoning. So you need to go backwards on a couple things. You need to sort of think about the answer and work backwards towards the question. You also need to think about, for page 6, what things are absolutely true, what things are likely true, and that'll kind of help you move backwards to solve those. So feel free to call each other and use each other for help on page 6. Again, it's meant to be challenging. We're not worried about grades right now. I just want to see how well you guys can solve that. Okay, remember there is a Zoom today after school, 2:30. Hope to see some of you there. Bye.